previous lecture, we have discussed about continuous and discontinuous buffer systems. Also, we have discussed about the STS page electrophoresis, which is a form of discontinuous system. It was developed by Lamelli, but before that Ornstein and Davis have developed the particular non-denaturing discontinuous system. Now, in this lecture, we are going to discuss some more techniques which are utilized for analysis of proteins. These are isoelectric focusing and 2D gel electrophoresis. Also, we are going to discuss about the detection methods, that is how the protein is detected in a gel, and also preparative electrophoresis, that though it is an analytical technique, but you can always purify a very high quality protein here by preparative gel electrophoresis. All right. So, we will begin our discussion with isoelectric focusing. Now, isoelectric focusing is an electrophoretic technique employed for separation of amphoteric molecules. For example, proteins are amphoteric molecules, and this is done in a pH gradient. It separates proteins based on their different isoelectric points. Proteins or any other amphoteric molecules, all amphoteric molecules will have a particular isoelectric point. Now, the net charge on an amphoteric molecule depends on the pH of its local environment. So, the amphoteric molecules, like as we will be discussing more about the proteins, we will focus on proteins here. They will carry a positive charge. So, any amphoteric molecule will carry a positive charge or a negative or a zero net charge depending on the pH of the environment or surrounding. Now, what is isoelectric point? Now, isoelectric point is defined as the pH at which a protein has no net charge, that is the number of negative and positive charges balance each other, they are equal, and this is a measure of the protein's net charge. So, the isoelectric point is a characteristic physicochemical property of every protein. Now, I think all of you know that proteins are positively charged at pH values below their pi, and they are negatively charged at pH values above their pi, and like the charge amount of charge will depend on how far they are from the pi value, or the pH surrounding pH how far it is from the pi. So, they will as they will contain a particular charge, net charge at particular pH depending on their pi values, they will migrate to respective electrodes in an electric field until the protein or the molecule focuses at the position in the pH gradient where its net charge is 0, that is isoelectric point. Now, proteins migrate to their respective steady state position where pH in a pH gradient equals pi from anywhere in the system. That means that even if you load the protein or amphoteric molecule anywhere in the pH gradient in the gel, they will migrate to its pi or isoelectric point where pH equals their isoelectric point. So, separating proteins according to their net charge is accomplished by generating a pH gradient in an electric field. So, first thing in isoelectric focusing is to create a pH gradient in the gel. We will discuss that. Now, the effect of protein size on mobility is minimized here, and to do that the electrophoresis is carried out in gels with large pore sizes. And if you could recall, the low concentration acrylamide gels like say 4% or around 3.5% or so, or say 1% agarose. So, large pore size gels, if you could recall, such as low concentration polyacrylamide gels around 3.5 or 4%, or you can use agarose gels which are 1% agarose gels. So, the large pore size minimizes the molecular sieving. So, and so those restrictive or impair the migration patterns are not impeded by the pore size here. So, this typically the protein will run on its charge. Now, how a pH gradient is created in the gel? A pH gradient is generated with synthetic carrier ampholytes. What are these synthetic carrier ampholytes? These ampholytes are a mixture of polyamino polycarboxylate compounds, and they carry different charges, net charge, and they are about very small, like 300 to 1000 Daltons in size. These are multi-charged organic buffer molecules with closely spaced pi values. So, and they have high conductivity. So, in a mixture of ampholytes, there will be lot of compounds with different closely spaced pi values or isoelectric point values. So, initially the pH of an ampholyte solution, if you make, will be the average of the pK values of the mixture, but on application of an electric current, what will happen is they will cause the ampholytes to migrate towards the electrodes according to their charges. So, ampholytes that have pK values above the pH value will be positively charged, and those with pK value below the pH will be negatively charged. So, now as the ampholytes migrate on application of electric current, this will result in charges in the local pH, this will result in the changes in the local pH due to the buffering action of the ampholytes, and this change in the local pH will affect the change affect the charge on the ampholytes. So, as they migrate, there will be changes in the local pH, and they will certainly affect the charge on the ampholytes depending upon their pKa values. So, the ampholytes will continue to migrate until they reach a position in which the local pH equals their pKa, that is no net charge, or you can say Pi value. So, the result is that a pH gradient in which the most basic ampholytes are found at the cathode, which is contains a dilute alkali solution like NaOH, and the most acidic ampholytes are at the anode, which contains a dilute solution of say H3PO4. So, you have what you have done is you have created a pH gradient in the gel. Carrier ampholytes with defined pH range can be purchased from different commercial providers, and they come in different pH ranges, like you can go from pH 3 to 10, or you can go from say 4 to 8, and different pH ranges are available, and they are commercially can be purchased from commercial providers. Now, proteins, if you say proteins also contains both, they are multi-charged molecules, they are also ampholytes or amphoteric, but they will migrate within the pH gradient until they reach a pH equal to their isoelectric point. So, problem with this is that concentration of ampholytes is higher, but proteins here cannot be used for creating pH gradient as such. In pH gradient created by ampholytes, mostly the concentration of ampholytes is about 2%, which is appropriate, and as concentration, if it is lower, that is say below 1%, it will result in unstable pH gradients, and above 3% ampholytes solution will be very difficult to remove from the gel, and may interfere with protein staining. So, usually 2% ampholyte concentrations are taken. Carrier ampholytes are are needed, since the protein concentration is generally not high enough to establish a stable pH gradient. So, proteins are not cannot be used as ampholytes, although we are utilizing this gel to analyze the proteins here. And they cannot give a stable pH gradient due to low concentration, and the isoelectric points of the protein may not be uniformly distributed along a pH gradient. So, in ampholytes you have a mixture of ampholytes with lot of different types with closely spaced pi values. So, they are more suitable for pH gradient. This figure shows in here, if you could see that you have casted a gel here. Now, this gel contains say lot of different ampholytes. There is a dilute alkali that is negative electrode, here it is a positive electrode, and as you apply the electric current, they will certainly migrate to their respective places and then they will concentrate there. So, that will be the way how pH gradient gel can be created. Let me show you on your screen. These isoelectric gels could be both, it could be simple tube gels as well as it could be slab gels. Now, what is done if in a it is in a tube gel or in a slab gel. So, first thing is that in a tube gel, for example, you have the gel in here. Now, this gel contains ampholytes. When you cast the gel, it already contains ampholytes all over the place. So, when you apply the electric current, then these ampholytes migrate to their respective places. As they move, the pH gradient is created, as we have explained, and so you can have say a pH gradient from say if these are 2 electrodes, then you can have pH gradients from say 3 to 10 or so. Now, so here you have a pH 3, here you have a pH 10, and it will mean you have a increasing pH in this order. All right. So, they will be like as they migrate and they settle down at their pi values, which you can then utilize it for this gel could be utilized for isoelectric focusing. So, what is done next? So, here what you have to do is then you have to load your sample protein. Now, remember protein could be loaded anywhere like I said, but it could be loaded on one side. You have particular pH here, a negative and positive electrodes. So, when you load the protein and as you apply the electric current, the protein will this protein sample will migrate according to their pi values, and they will migrate towards or they will settle down, or you can say focus at particular site where their pi values where pH equals their pi value. So, when you apply the electric current, then what resultant you will get is that you will get different different bands which could be visualized by staining actually. Same will happen in a slab gel, like a slab gel, like for example, I can just show you here. In a slab gel, say there is a negative positive electrode and say the pH is there is increasing pH here. So, if you have say pH starts from now you have a protein which you can have either load here, same protein if you can load here, and if the protein pi is say around 6, then this will migrate towards this side and this will migrate towards this side, and finally, they will settle down at pH 6 or they will focus at a particular pi value. So, this is how you can focus, you can see that because of the pH gradient due to ampholytes, the proteins loaded anywhere will come and focus at a particular place in isoelectric focusing. So, that is why it is the name is so. All right. So, let us return to our discussion. So, isoelectric focusing is a very high resolution method, and it can resolve proteins which are differing in pi values, say around or less than 0.05 pH units. So, even if they are spaced by 0.05 pH units, then they could be separated on isoelectric focusing gel. The technique is performed in non-denaturing conditions most of the time, and it is quite rapid. Like for example, proteins, say enzymes, antibodies, antigens or other proteins, they will retain their activities in this non-denaturing conditions. But many times denaturing agents could be added, but those denaturing agents like they could be non-ionic detergents or urea can be utilized. This happens in certain cases, particularly like say proteins may precipitate near their pi value, or you want to solubilize membrane proteins, then you might require addition of say urea or non-ionic detergents. Even in two dimensional gel electrophoresis, these non-denaturing agents will be utilized. So, this method can be analytical, like it could be just for analysis and not for purification, as well as this method could be preparative also. Now, typical analytical gels take around say 90 minutes to say 2 hours or so to run, depending on the voltage conditions, whereas preparative gel may take more time, say around 4 hours to complete. So, here as we have seen that the main points or in IEF is that you have to first make a pH gradient gel, and in that pH gradient gel you can run your protein samples which carries a particular charge at particular pH according to its pi value, and then it will migrate to its pi value in the gel and it will focus there. For example, if say if diffuse is little bit ahead or it is remaining behind, then also after some time it will focus on there. Like if it is diffused a little farther, then it will come back and focus. So, it is a very good technique, it is a high resolution technique. Now, like I was saying, it could be both analytical as well as preparative technique, like you can use it for just for analytical purposes, or you can purify the samples. In analytical IEF, it is mostly carried out in large pore acrylamide gels, and these are like 5% T and 3% C. Hope you remember what is T and what is C. T is the total gel concentration, like acrylamide concentration, and C is the cross linker percentage in total concentration of acrylamide, that is bisacrylamide. It could also be run on agarose gels like 1% agarose gels. So, traditionally earlier 1 to 2 millimeter thick gels were used, but then high cost of ampholytes makes the procedure very expensive. So, what was done? Introduction of thin or ultra thin IEF gels was proposed, were introduced, and these were like say around less than 0.4 millimeter or around 0.15 millimeter gels were introduced, and they have considerably reduced the cost of running IEF gels. To prepare these ultra thin layer gels, what is done is that carrier ampholytes and it will be of suitable pH range as per your requirement, and so you have to take care that your protein pi lies in that particular pH range, and it could be suitably if it is a mixture, it could be suitably isolated and differentiated. So, carrier ampholytes will be mixed with acrylamide solution and initiator system. So, what is initiator system here? Polyacrylamide gels here in IEF are polymerized by photopolymerization and also chemical polymerization, but so here initiator system includes riboflavin, which could be around 5 microgram per ml. If you could recall, riboflavin is used for photopolymerization rather than like free radical. So, here you have riboflavin, then you have ammonium persulfate also and timid also. Ammonium persulfate around 0.015% and timid 0.05%. Now, why we are why these all these three uh, uh, chemicals are used here? It will result in more complete polymerization, and it has been seen when all three initiators are present, then the polymerization is much better. So, the polymerization takes place for almost more than 2 hours, and where in second hour particularly direct light from a fluorescent lamp or other source, the gel will be exposed to that light, and so photopolymerization. So, you can say photochemical polymerization can occur in here. So, this is done in IEF gels. Now, for most analytical isoelectric focusing, the horizontal polyacrylamide slab gels are used. Even in agarose gels, their horizontal gels are utilized, but polyacrylamide is most commonly used. Now, gels are cast on a glass plate, or they could also be cast on specially treated plastic sheets. Uh, Now, it's a laborious, and you have to work very carefully when you are casting these gels. So, many times these pre-casted gels or gel strips with different pH ranges are available commercially. So, you can directly buy. Of course, it will be expensive here, but it is very hard to you can make gels, but it is very hard many times to reproduce the results, which could be done in commercially available gel strips or gels. So, once you have casted this on a glass plate, like we have said, you have to mix all these things like acrylamide, ampholyte solution, initiators, and then it has to be polymerized. Once it is polymerized, as we have discussed earlier also, it could be polymerized taking 2 plates and then 1 plate is removed, or in a different way. Then electrolyte strips are saturated. Now, these electrolyte strips are saturated for both anode and cathode. So, one like 0.1 to 1 molar phosphoric acid is utilized for anode, and NaOH, which could be from 0.1 to 1 molar NaOH is at the cathode. So, you have positive and negative electrode buffers, you can say, and they are placed on the gel and they are in contact with the power supply. So, they are these electrolyte strips make contact and they are maintained, this contact is maintained by electrodes of platinum wire. Now, IEF can also be done in tubes, like I was saying. You can do it in slab gel, but you can also do it in tubes, and this constitutes if you are doing in tube gels, tube gel also constitutes the first dimension of 2-dimensional gel electrophoresis. After the gel is cast, a potential difference is applied, which allows the formation of pH gradient between the anode and cathode. So, what is done? Once you have made, you have prepared this all this apparatus, and this particular arrangement is made, then first thing is the casted gel contains ampholytes, they are scattered just like that. So, a potential difference, say for some time is applied, say for 30 minutes or maybe around that time, and the ampholytes will migrate to their respective Pi values, and this will create formation of pH gradient will be done between the anode and cathode. So, that is the first part of running the gel. Once the pH gradient has been made, then sample could be loaded. Now, suitable samples can be prepared by dialysis or gel filtration, and you have to see what is the chemical buffer concentrations, other things has to be looked into. So, they could be prepared by dialyzing for suitable buffers. Now, samples once pH gradient is made, samples can be applied to these thin IEF gels. In tube gels, you will be applying it to one end actually. but in like say thin IEF gels, they are directly placed by, you can, what you can do is you can soak the filter strips in the sample, and they will, when you are soaking, they contain enough sample, you make sure that they contain enough sample, and these buffer strips containing the sample could be directly placed onto the gel. Uh, Alternatively, one can also load, say, a few microliter of sample can be placed directly onto the surface of the gel. Maybe there might be a few cassettes sort of where you can put the gel, where those things could be put on either in the middle of the gel or on one side of the gel, and then once you have loaded the sample either through buffer strips or directly a few microliter onto the gel in certain cassettes. Voltage is again applied, and for about say 30 minutes or so, and this, what it does is it allows samples to electrophorese out of the paper or from those cassettes into the gel. Uh, so, applying voltage makes sure that your sample is loaded onto the gel. Now, to achieve rapid separations, uh, then once you have loaded the sample, you can run the gel for like say at a particular voltage, and like I said, to achieve rapid separation, relatively high voltage are used, and when you use high voltage, certainly the heat generation will take place. So, gels are run on cooling plates to minimize this heating. So, once gel is run, then the gel could be visualized on by staining, like say if you are staining it with Kamasi blue stain, then around 0.5 microgram of protein is required for dye staining, and 50 nanogram of protein per band is required with silver staining. That is the sensitivity range of these methods. Now, staining solution for proteins in IEF, they consists of 0.04% Kamasi brilliant blue R250, or with containing 0.05% crocin scarlet in 27% ethanol plus 10% acetic acid. Gels are soaked in staining solutions for 1 hour, and then staining is done by with several changes of 40% ethanol and 10% acetic acid. Silver staining can also be done for higher sensitivity. So, let me just show you a little bit that how this whole thing was done actually. So, what is done is, like, you have a very thin gel of certain size which is there, and this gel will be, there will be like wicks or paper filters like I said, where you will have phosphoric acid and NaOH at different electrodes, and so they will be connected here like all over here through platinum wire to the power supply. Now, loading the sample could be done like you can have buffer strips with thin buffer strips, and these buffer strips are soaked in the sample solution. So, you can soak these buffer strips in the sample solution, and then they could be put say on one side, or many times you can put them in the middle also. Simply you can put them in the middle of the gel, or you can put them on either side, and this is a pH gradient gel, say we say 3 to 10 or different ranges could be utilized. Now, what is done is, this strip contains the solution and this remains in the here, and as you apply the voltage, then the from the strip or from the this buffer the filter strip, the solution will get into the gel, this thin gel. There could be another ways, like what you can do is, rather than having this particular buffer strip, there could be simple obstacles you can put in here on both sides, and samples in a very small amounts, like you can have separate, like say you want to load more than one sample here, then there will be like slots sort of here. Now, here remember these are not wells as such actually, rather these are slots which you are making it the physical material you have put in, like say cassettes sort of, or empty cassettes, or hollow kind of plastic cassettes kind of, and then sample could be loaded in here. So, you can load your samples separately in each of these slots, and then it could be run, and you can perform the run, and as you run it for 30 minutes, this will electrophorese into the gel. All right. So, this was about the gel analytical gel electrophoresis. Likewise, preparative isoelectric focusing can also be done. So, what we have seen till now, analytical isoelectric focusing, which is done on ultra thin or very thin gels or in tube gels, and where you are not really aiming to purify the samples, but in preparative isoelectric focusing, you will be working on like spatial cells or rotophores which are utilized, and they could be like in solutions or otherwise, and large amount of proteins can be like could be separated in that, and could be subsequently used for different applications. So, we are not going into details of that, but it is possible in isoelectric focusing to purify the protein, and you get very good quality, that is in terms of purification, like homogeneous sample, and you can utilize it for different applications. All right. So, now we move on to the next technique, that is two dimensional gel electrophoresis. Now, this technique combines the technique of isoelectric focusing, which separates proteins in a mixture according to the charge, and then next will be the size separation technique at 90 degrees. So, now remember two dimensional gel electrophoresis means any technique which combines two runs at 90 degrees or at perpendicular to each other. Like you with one technique you run it in one direction, and then you same gel could be run at 90 degrees, like you turn or in perpendicular direction, but most widely used combination is the isoelectric focusing and STS page. So, the two dimensional gel electrophoresis which we are going to discuss about is about this particular one, that is first the separation of proteins occurs on isoelectric focusing on the basis of their pi values, and then they are run on at 90 degrees on STS page gel. Now, the first dimension, that is isoelectric focusing, is carried out in polyacrylamide gels in narrow tubes, and this could be say 1 to 2 millimeter internal diameter in the presence of ampholytes, like I showed you that first you have to create the pH gradient and then run. Here you will use denaturing conditions where 8 molar urea and non-ionic detergents could be used. Now, the denatured proteins separate here according to their isoelectric points, and once that is done, then the gel is extracted from the tube. You can just apply slight pressure to one end and it will come out. Then it is incubated for 15 minutes in a buffer containing STS. Now, this what happens here is that binding of STS to the denatured proteins occurs here, and then they are placed along the stacking gel. So, you have casted the STS page gel, which is discontinuous gel. So, you will place this tube gel which was in tube and which has separated the proteins on the basis of their pi values. You will just lay it on the stacking gel, and what is done is you will pour some molten agarose here over the gel in electrophoresis buffer. Now, once the agarose has set, then electrophoresis is started, and the STS bound proteins will run into the stacking gel and then separate according to size in resolving gel. So, that is a very simple way of running a two dimensional gel. So, what you have here is, if you can see here in this figure, first thing you have done is isoelectric focusing has been performed, and then in isoelectric focusing you can see various bands here of proteins, which is you can see after staining. If you do not want to stain, that is also all right, but one time you can see that it is doing well. Then what happens is, you will lay this gel and it will run. Now, you have run in this direction and now it will run in this direction, that is at 90 degrees to the previous run. So, you have put it on a stacking gel and then it will run. It could be a gradient gel or step gradient or continuous gradient gel, where your pore size will decrease as you go along. And what will happen then? The proteins will run as per their. So, one time they have run on the basis of charge, now they will run on the basis of their size. So, smaller molecular weight proteins will run faster and larger molecular weight proteins will remain on the top. So, that is how you will be able to run the same sample in two directions, and this gives now what is the advantage of this is that you can handle lot of proteins at a time. Like say you would like to do the protein analysis of a particular tissue sample or cell sample, or you want to see the expression of a particular protein and you can compare the native one and where you have put in cloned a new protein in a particular system, and you want to see the expression, then comparisons can be made and the extra band which is shown of particular protein could be made out. Or likewise, in certain diseased conditions, the normal cell and diseased cells could be compared for their proteomes, that is total proteins, and then again it gives a lot of information. Likewise, unknown protein samples from a particular system could be analyzed. Now, here you can see this is how a 2D gel will look like after the two dimensions, and you can see there are a whole lot of bands protein like spots here. Now, these spots, remember, there could be many a few thousand spots here which can be handled at a time, and this protein spots you can pluck the spot and this could be analyzed on mass spectrometry, which can give you the sequencing results and you can identify the protein in here. So, this is a you can say high throughput procedure to analyze a lot of proteomes from different systems actually, and a very useful technique in combination like IEF and STS page. So, two dimensional gel electrophoresis is one which is a very good technique for, and it is widely used with mass spectrometry. All right, now when you have run these proteins on different kinds of gels, now let us little bit discuss about their detection methods. So, the proteins separated in gels are detected mostly by staining with dyes or metals. Kamasi Brilliant Blue R250 is the most commonly used for routine work, but Kamasi Brilliant Blue G250 is also used for staining low molecular mass polypeptides. Then silver staining is the most sensitive method for proteins and as well as nucleic acids, and is used for assessing high purity of the sample. Then there is also another copper staining which is rapid and sensitive method for STS page only, and where proteins are not fixed. So, you can elute also protein. There could be other methods like fluorescence and radio labeling. Now, all methods are performed with precautions, taking care. They are done at room temperature with gentle agitation or shaking on orbital shaker, and the gel and staining solutions are contained in a glass container or plastic container. Now, Kamasi blue staining is a very popular method and a very routinely used method. So, what is done here, staining solution consists of 0.1% Kamasi Brilliant Blue R250 plus 40% methanol and 10% acetic acid. Now, this methanol and acetic acid also fixes the protein in the gel, that is precipitates the protein in the gel. So, that it is fixed there, that is why it is called fixing the protein. And after electrophoresis, gels are incubated with agitation in staining solution for 30 to 60 minutes. Then after that, excess dye is removed by destaining the gel in the acetic acid and methanol solution, same percentages with several changes, so that a background dye is gone, and then you can clearly see the protein bands. Gels containing low molecular mass polypeptides are fixed in 50% methanol and 10% acetic acid for 1 hour, and then stained with 0.025% Kamasi brilliant blue G250 and 10% acetic acid, and then destained with 10% acetic acid. So, for smaller molecular weight polypeptides, G250 is more common. In terms of sensitivity, protein bands containing 0.1 to 1 microgram could be seen in here in Kamasi brilliant blue staining. Then silver staining is almost like 100 times more sensitive than Kamasi blue staining. It can detect like 10 to 100 nanogram of protein or nucleic acid per band in a particular band. Method is based upon binding of silver ions to proteins and reduction of the silver ions to metallic silver. So, fixation of proteins in gel is performed with methanol and acetic acid, and then methanol is removed by washing with ethanol 10% and 5% acetic acid. Here then proteins are oxidized in a solution of potassium dichromate in dilute nitric acid. Then excess oxidizer is washed off the gels with water, and then gels are treated with silver nitrate solution. So, the silver ions bind to the oxidized proteins, and then they are subsequently reduced to metallic silver by treatment with alkaline formaldehyde. So, color development happens, and that could be stopped with 5% acetic acid after some time, otherwise lot of background might or a lot of diffuse bands might be seen. Another method for silver staining is available in a kit form, and it requires single staining and development step and could be used for both proteins and nucleic acid. After fixation in 50% methanol and 10% acetic acid plus 5% glycerol, and after washing with water, gels are treated with a solution containing silver amine complex bind to tungstosiliceous acid. Then silver ions transfer from this to proteins or nucleic acids, and then they could be reduced to metallic silver with formaldehyde. So, silver ions do not accumulate in the gel, and that gives the less background. Again, you have to stop the color development in proper time. Photo development can also be done, light could be used to reduce the silver ions to metallic silver. So, this is how the gel will look like in when you have silver staining, and like I said, it has a very high sensitivity or the highest sensitivity, much more than the Kamasi brilliant blue staining. Then copper staining is a very rapid single step staining of only SDS page gels. Staining is performed by incubating gels in 0.3 molar copper chloride for 5 minutes, followed by washing with water. So, what will happen is that blue green precipitates of copper hydroxides in the gel will be seen, except where there is high concentration of SDS. So, where protein bands are there and the protein contains lot of SDS. So, there copper will not these blue green precipitates will not be seen. So, against a black background you can see clearly the protein bands, and these proteins are not permanently fixed. So, they can be eluted here, but problem is that electrophoretic patterns will not last for long. So, you have to when you dry the gels, they will be gone. So, either you should store them in water or photograph it right away, or you can restain with Kamasi blue. Sensitivity is somewhere in between the Kamasi blue and silver staining. So, copper staining is also a very good method where you want to do preparative electrophoresis. There are other methods also, like fluorescent dyes that binds non-covalently to proteins, and they could be utilized and like say through UV illumination or others. You can use fluorescence dyes. Also you can attach, but that will affect their charges. Then autoradiography could be used or fluorography could be used. As if you can remember, we have discussed this in a section where we have discussed autoradiography and fluorography. Like in gels, you can put radioactive material, and then through autoradiography they could be seen. They could be enhanced by fluorography, where exposure of film to secondary light emitted by either intensifying screens or floors, etc., could be used. So, these methods could also be utilized, but they are not so commonly used methods. Enzymes can be detected on the basis of their activity, like after running the gel, but it should be non-denaturing gels. Substrate could be added, and the reaction happens, which can form insoluble products, and that could be seen here. Like in, for example, in proteases and others, this could be utilized. This is how it looks like when you are doing those gels. Activity patterns could be seen through different methods of staining, and we are not going to discuss those things in here. These are gelatin, the gels contain gelatin, and like for example, in proteases, case of proteases, they can cleave certain part of gelatin, and clear bands could be seen in here. Quantifications as such is not possible, but quantifications in the gels could be done by comparing it with the standards. Like you can load known amount of the sample, and then unknown protein could be compared by visualization or digitally. And so, that could be a method to like by generating standard curve, you can really quantitate it. Also, radioactive proteins can be excised, and then on liquid scintillation counting, that could be done to quantitate the protein. Scanning densitometers is usually convenient method to quantify the particular amount of protein. So, these are few methods of quantification. Now, little bit preparative, like this electrophoresis technique, like I said, is more of a analytical technique, but you can use it for preparative electrophoresis as well. So, here preparative electrophoresis, this could be two methods. One is that you elute it from the gel, and or another would be that you can do continuous flow electrophoresis. So, in elution, like you can do it by two methods. One is the simple diffusion or passive diffusion method, where what you do is you can cut the gel, the band from the gel, that is protein band or nucleic acid band, and you can macerate it, and you can leave it in the buffer, and the protein will diffuse into the solution. But the recovery is very low and process is very slow, then protein is in very diluted form. Other way is electroelution, that is you force the protein through electric current to migrate to one side and out of the gel. Like say you can put them in a dialysis bag and then can run and then can apply electric current, and they might come out of the gel because they have to run towards particular electrode, particularly for nucleic acids. Then there could be continuous flow electrophoresis, where it could work like a column, that you continue the electrophoresis until all of the proteins migrate off from the bottom of the gel. If you could remember, we have to switch off the gel when you are having say bromophenol blue or dye reaches the bottom, but here you allow it to go through. In the chamber, it is sealed with dialysis membrane to prevent the proteins migration into the tank buffer, and then that could be even you can put UV monitor or fraction collector, those things could also be done in here. Remember, these gels for preparative electrophoresis are not thin gels as we are utilizing, they are much thicker gels, so that more amount of protein could be loaded and good amount, and you can do it in like this one could be done in tube gels, continuous flow, and thick gels could be cast, and a lot of protein sample could be of high quality could be purified in there. As it's the purity is quite high in here. This is a simple thing, like you have a gel and there is a buffer reservoir here, but there is dialysis, like you collect this and this will be directed to a pump or monitor, and then this is how you overrun the gel, but of course, you do not allow it to pass to the into the tank buffer. So, this is how preparative electrophoresis can be performed, where you can recover lot of samples actually. So, we can summarize today's lecture here. What we have gone through today is, one is isoelectric focusing, which is a very important technique, and here you can purify or you can separate proteins which are differing from say 0.05 pH units, and that is quite good. And on the basis of isoelectric points you can separate these proteins and analyze them. Even you can purify these proteins in preparative isoelectric focusing. pH gradient is made through ampholites, and then you can once pH gradient is made, proteins migrate to the particular pH which equals their pi, and they focus on there. And so, this could be utilized for both analytical and preparative purpose. 2D gel electrophoresis, the most commonly used is, which combines IEF and STS page, and it is widely used for analysis of proteomes like proteins from a system. Even comparing say two systems with minor differences like say overexpression of a protein, or normal and diseased cells, or otherwise it could be utilized for a lot of different applications, and it is combined with mass spectrometry. Also, we have seen the detection methods where Kamasi brilliant blue or staining or silver staining or copper staining could be done. With silver staining is the most sensitive of the three, and preparative electrophoresis is one other method, though chromatography is more common because you can use purify large amounts, but in here also you can purify the sample with a very high purity through preparative electrophoresis. So, in the next lecture we will move on to the electrophoresis of nucleic acids. Thank you.